today's lecture. So in this lecture, I'm going to be talking about how do we assess the quality of our raw read data. Previously in the previous lecture, I spoke to you about the overall data analysis process going from raw reads, so FastQ data, to variant calls in terms of VCF. So I'm going to go over some of the material from that just to recap. But before I start with that, I just want to go over some of the objectives for this lecture. So at the end of the lecture, we hope that you'll be able to understand the FastQ file format, understand the principles underlying the quality control of FastQ files, describe the output produced by FastQC, which is a program that we use to assess quality, understand and interpret the quality control metrics, so the output files generated by FastQC, and also understand how to trim FastQ files to improve our data quality. So the first couple of slides are again from the last lecture, just to recap. So in terms of the data analysis pipeline, we are talking about the bioinformatics analysis, and we're starting off at the beginning here, where we get our read data in FastQ format files. This is what we call our raw data. And what we're going to be looking at in this lecture is this component here, where we run the FastQ data, FastQ files through a program in order to assess the quality. We get these quality control reports out in HTML format, which we then look at and we assess. And based on the quality that we observe in our data set, we then decide if we need to trim. To trim our data set, we can use the tools to trim that, and we trim our FastQ data set, and we remove sort of low quality reads or regions of our reads that are low quality. And that is the input that goes into our next step, which is either assembly or alignment. Okay. So again, this is just a recap. So we're starting off here at the top. This is the first component of the data analysis pipeline. The FastQ format we spoke about before, so hopefully everyone's familiar. I just wanted to put in this slide. So probably many of you have seen Sanger sequencing before. So this is an example of an output from a Sanger sequencer. And essentially this is the reads that come out. And I just wanted to make the comparison. So for example, if you look at this region here, TCAT, these peaks and how high these peaks are essentially what is the equivalent of our quality scores that we observe in our FastQ data. So the higher the peak and the more clear the peak that comes out, the more confidence we have in these calls. And in the same way, the higher our quality scores are, the more confidence we have in our reads that are our bases that have been called from our next generation sequencing experiment. This is an example of the FastQ files. So essentially, I mentioned this before, that when we get our data back from a sequencing experiment, we'll usually get 2 files per sample. If we've done paired end sequencing, if you haven't, there might be 1 file. Some providers will give you both the forward and reverse reads in 1 file. That's not standard practice. Usually you do get 2 files separately. And again, here you can tell which are your forward reads by the number after the slash. So number 1 is indicating it's a forward read. If we had number 2 here, so our second file would, for this particular sample, would have 2 here indicating that that's the reverse reads. So for a single sample, we'll get 2 files. We'll get a set of reads on the forward from the forward reads, and then a set of reads from the reverse reads. Okay, a set of sequences from the reverse reads. Okay, and we obviously have to assess the quality of both of them. This is just to recap. We have the Fred quality scores. So the quality scores will give us some indication of how likely it is that that base call has been called due to an error or is an error. And we basically consider something with a Q score, quality score of 30 as being a reasonably high quality. That Q30 is the equivalent of the probability that that is an error is 1 in 1,000. And then this is just about the coding and the percentage, and this is another way of looking at it in terms of confidence. So again, the Q30 that we use as a cutoff, we're essentially saying that we are 99.9% confident that that is a true call with a 0.1% error rate. So the chance of it being an error is 0.1%. And we have this trade-off between accuracy and error. And in general, we use a Q20 quality score of 30 as our threshold. So now we're going on to some of the newer material. So why do we need to do quality control? So the major reason behind this is that errors can happen at multiple steps during the sequencing process. So errors can be introduced into your data during the library preparation as a result of how your library was prepped. It can be introduced into your data as a result of the quality of the DNA that you used. So generally, low quality DNA will result in low quality sequencing. Then errors can be introduced if you are not doing a PCR-free sequencing approach, which is normal Illumina sequencing, where we do have a PCR step. Errors can be introduced into your data during PCR sequencing, PCR amplification. And then obviously all sequencing technologies and chemistries have certain error rates that are associated with them. So for example, some sequencing technologies struggle when they are homopolymer regions, for example, where you have strings of a single base, so strings of A's or strings of T's. The sequencing technology might not be able to identify whether it's 5 T's in a row, whether it's 6 T's in a row. There might also be other complex regions of the genome where sequencing technologies will fail completely. So there's a number of different stages at which errors can be introduced into your data set. And essentially at the end of the day, when you get out your variant call file, your VCF file, you want to be sure that you've been able to remove as much of those regions where we have questionable calls, so that you don't end up with what we call a lot of false positives in our data set. So variants that are identified as variants in our samples, but are not real variants. They weren't actually there when we started off with our DNA. They were either introduced somewhere along the line during the sequencing process, or during the sequencing process, that base was called incorrectly. Okay. So what we do know is that we know a lot about the biology, so associated with, for example, the transition versus transversion rates. We know a lot about complex regions. We know a lot about repeat regions. So we know a lot about biology and where these errors might be more likely to exist or most likely to occur. We also know about the sequencing technologies, and we know that certain sequencing technologies are weak in certain regions of the genome. They might not be able to call homopolymers accurately. So we know a lot about where these errors are likely to occur. And essentially we can use that information to try and identify where these sort of low quality bases might be called. Okay, so the idea behind this is just to realize that although we are sequencing, starting with DNA, and we're doing whole genome sequencing or exome sequencing, there might be errors that are introduced into the data sets. And so the quality control step is an important step to do. And what you'll see is also at the end, we also can then filter out our results to again remove low quality calls that are most likely to be false positives. Okay, so errors can be introduced at various steps. I mentioned this before. So during library preparation, we might have some errors being introduced into our genome, and we might, depending on how the libraries are prepped, that might affect how we actually generate the quality of the sequencing that's generated. We have some errors that are due to the sequencing technologies and the chemistry. And then we have some errors, again, that might be introduced during the data processing steps. So when we actually are processing our data, we might have algorithms that are not capable of dealing with some complex regions that might introduce some errors into the step. For example, when we're mapping, say we're generating region, we're mapping back to a reference, I showed you in the previous lecture where we have these regions that are repetitive in the genome. So we might have instances where the algorithm is not able to accurately map those reads to the correct regions when we have repetitive elements. So the QC part of the NGS analysis pipeline is the first step, and it should be conducted with caution. But the other thing is that you must do quality control on your data set. I previously used to come across people who were like, no, we're not going to quality control the data because we need to use as much of the data as possible because we only have a few samples. And that just doesn't cut it anymore. If you don't quality control your data, at the beginning, any result that you get out is questionable. So one of the characteristics that we know in general is that our NGS read quality, so for a single read, it usually starts off with high quality and the quality drops off towards the end of the read. And this is essentially just due to the way that the sequencing chemistry works. And some of the platforms are better at this and some of them are worse. So you'll find that the quality might drop off dramatically on some platforms, but on some platforms it might be better. And we'll see an example of that in the lecture. So there's various other, so the one thing that we do look at with the read quality is obviously looking at the quality scores. That's the obvious one, because we have a quality associated with every single base that's being called, right? So the one thing we can assess straight away is in a particular read, we can look at the quality that corresponds to each of the bases, right? And as I mentioned before, a threshold, the general threshold that's used is if you have a Q score of greater than 30, we're pretty happy that that's a high quality call. Okay. There's also various other quality metrics that we can use. So overall, across our entire data set, we can look at a number of other quality metrics, and then we can use that to assess whether our data looks like it's acting normally, or we're getting results that we expect to see. Usually, if we get results that are outside what we expect to see, we can then say, oh, well, it looks like there's something going on, something strange going on with my data set overall. So that's something that we need to be aware of. And then we can dig deeper to try and find out what the problem is. So a very common tool that's used to assess the quality of whole genome sequencing FastQ files is a tool called FastQC. There are some additional tools. For example, there are tools within SAM tools that you can use to generate a number of quality control statistics and metrics. It's a much more manual step approach with which you can run on the command line. So if you are interested in that, just have a look at SAM tools and quality control. But generally what people mostly use is FastQC. The reason for this is that it's quite intuitive and simple to use, and it generates a report with various metrics and graphs in HTML format. Obviously, anything visual is much more easier to interpret. And so what you do is you can run FastQC is a command line based tool. There is also a graphical user interface for it. We'll be using the command line version. So you can run a FastQ file. So you'll have, say, for example, for sample one, you'll have your FastQ file for your forward reads in a paired-end experiment. You'd run that FastQ file through FastQC. It'll generate a single report based on that sample one forward reads file. You'll then run the reverse reads. It'll generate a single report for that. So that's essentially how you analyze your FastQ files. So for each FastQ file, you will get a report telling us information about that FastQ file. Obviously, you start to think about this, that this might become a bit difficult to assess or to deal with when you're dealing with thousands of files. So there are some automated ways of doing this. There are some automated ways of processing the reports files, but we're not going to talk about that here. What I am going to mention is that there is a tool called MultiQC, and MultiQC is basically a visualization tool. And what it's able to do is you generate your FastQ reports with FastQC. So you have your FastQC HTML files. And what MultiQC does is it takes as input, say you've got 20 FastQC report files, you can input that using MultiQC. And what it does is it creates nice visualizations where you're able to look at all your data together. Okay, so that's a good tool to use if you're looking at multiple samples. Instead of looking at individual FastQC reports, you can then use MultiQC to import all those reports, and you have some cool graphical user interface tools that allow you to then assess the quality across all your files in one go. Okay, so for the sake of the practical, we're not going to look at MultiQC, but we are going to look at FastQC. Okay, so I'm gonna just pause there quickly to see if there are any questions. You're welcome to probably raise your hand and ask the question. That's going to be the easiest. Okay, no questions. Great. Okay, so now what I'm going to do is I am going to run through the FastQ report file. So this is actually a report file from an example dataset that's been run and this example dataset is actually available on the FastQC webpage. And I'm going to go through each of the quality metrics that are produced based on your QC file. Okay, on your FastQ file. So as I mentioned before, FastQC is an easy tool. You actually just on the command line. You guys have been using the command line yesterday. You just have install. If you have to install FastQC, you just say FastQC and you give it the name of your FastQ file. So FastQC sample1.fastq. Okay. You can give it multiple files as well. So you can say FastQC sample1.fastq, sample2.fastq, sample3.fastq, and it'll run it on each of those FastQ files. Okay. So the first thing that we will do, we'll look at now is the report. So when you run FastQC, it'll generate a set of files. One of those files will be a .html file. The .html file is a file that you're able to open in a web browser. Okay, so if you are generating, if you are running FastQC, for example, on a server, you will generate the report files. You will have to copy those report files. If you don't have a graphical interface set up on your server, you will have to copy those files back to your computer or anywhere where you have access to a graphical user interface. You have access to a web browser. So you won't be able to view the reports easily on the terminal. There are ways to do it. But if you don't have access to a graphical interface on your terminal, then you actually have to copy those files back to your computer in order to open them in a browser. Okay, so you'll copy them back to your computer and then you'll be able to open them in a browser. So when you run FastQC, the major report that you look at is the HTML file, and that is going to open up in your browser and it's going to give you a whole list of graphs and these different metrics. Okay, so we're starting at the top of the file. So essentially, this is on the right. Oh, I think it's the right-hand side or the left-hand side. You'll see the FastQ report summary. So these are all the different sections of the report. Okay, and we're going to go through these one by one. So at the top of the report file, you'll find the first section called Basic Statistics. And this is basically just looking at your FastQ file and generating some basic information about what is contained in your FastQ file. Okay, so here's our example. So we were running FastQC on a file called goodsequence_short.fastq. Okay, It'll tell you what's the file type. So it says your conventional base calls. It'll tell you the encoding. So remember the encoding was FRED33 I spoke about in the lecture. It's Illumina 1.5, which is similar to FRED33. It will tell you the total number of sequences in your file. So in this file, we had 250,000 sequences. It'll tell you if it filtered any sequences, so if it found any strange sequences that didn't look like proper reads. It will then tell you what is your sequence length. So this would usually be what you're expecting your read length to be. Okay, so in this particular data set, it's a small read, so it's 40 base pairs. Okay, again, this is depending on what experiment you're running, whether you expect it to be 150 base pairs, 100 base pairs. So this is also dependent on what experiment you run. And then it tells you the percentage GC, which is 45. Okay. So just to mention, your read length, again, is based on your experiment. So you must make sure that this read length corresponds to the read length that you're expecting based on your experiment, whether you are doing 150 base pairs, paired end reads, or something larger or something smaller. Okay. So this is essentially just the basic statistics that you get back on your FastQ file. Okay. The next page, well, the next part of the report, essentially if you scroll down, is the per base sequence quality. Okay. So this is the range of quality values across all bases of the read at each position. So remember before I said that we have, in this particular file, we have reads that are 40 base pairs long. Okay, so 40 bases long. Okay, so essentially what they've done here is that you'll see it goes on the y-axis. You have values from 0 to 38. This is our quality score. On the x-axis, we have positions in the base. So we have from 1 to 40. Okay, so what it's done is it's looked over all 250,000 reads, and at each position, it's plotted the quality score distribution of that position, all right? And it's plotted it as a box and whisker plot, okay? And I'll show you in the next slide what a box and whisker plot is. But essentially what it does, so for position 1, it's looked across all 250,000 reads. And what it's done is it's plotted the quality scores at position 1 for all 250,000 reads, okay? And this is what it's showing you here on top, okay? So basically, we're gonna go into this in a bit of detail, but this is the distribution. So the box, the yellow part is the distribution, and these are what are called whiskers, or the whiskers of the distribution. Okay, so our quality scores range all the way from 35. For example, for position 2, it ranges all the way from 35 to 38. And you'll see majority of our reads, the quality score is basically between 37 and 38. Okay, this is color-coded, and we'll come back to this a bit later, to give you some indication of what we consider to be good quality. So you'll see we go into green around 28. 28 and up is green because we consider that to be good quality. This is sort of in between, and these are what we consider below 20 to be low quality calls. Okay, so yeah, just to mention again, sorry, my so just bugging me a bit. So again, so for example here, if we look at, this is position 32. Again, it seems to be quite consistent that the lowest quality score that we recorded or we see in our data is about 35 at this position. And again, majority of our quality scores range between 37 and 38 and up. Okay. So this is explaining the box and whisker plot. So in the box and whisker plot, you're not seeing the full plot here, but essentially this is what a box and whisker plot looks like. So it gives you the range of the quality values across all bases of the read at each position. So remember I said we look at position 1, or let's say position 10. We say at position 10, if I look at all my reads, what is the, and I then look at each read and I plot that into the box and whiskers plot, and that becomes what the distribution is. So this is what the plot looks like. So these whiskers are basically giving you, remember I told you the minimum quality score we get is the bottom. The maximum will be at the top. In this case, we don't have the top of the plot because all of our calls basically fall into these quantiles here. Okay, so we don't have the top of the plot, this part here. So essentially, this is the minimum and the maximum you'd see at the top, and then this is majority of our samples will fall into this region here. Okay, you don't have to know about the lower quartile, upper quartile, but this is just showing you what these box plots, the box and whisker plots look like. So it's showing you, the box is showing you where majority of our quality scores fall. We have a minimum, that's the minimum quality score that we observe at that position. We have a maximum, and that's basically all you need to know. Okay, so going back to the good, acceptable, bad. So again, I told you that this is color-coded. So this is just to allow you to more easily see, to visualize the quality of your data. Ideally, we always want to see data where our reads, our box and whisker plots are falling into the green region. What you might have is that the distribution of the, so that your boxes might fall into the green region, and you might have some of your whiskers falling below that. That is fine. It depends on what you're accepting as high quality data. You might want to trim then based on that. But essentially, we always want to see our quality scores in this green region. Okay. So that's the first major quality control metric that we look at in our data. Okay, so here's an example of data that's a bit more messy. So in this case, you'll see that our data starts off reasonably well. We have data in the sort of, so this is 0 to 40, and this is a longer example of a longer read. So here we're looking at a read that's 240 bases in length. And you'll see it starts off good, and then we have this systematic sort of decline in quality across our reads. Okay. And this is sort of a good example of something where something happened with the sequencing with this particular read, where it started off good, and then towards the end of the read, it started failing, and our quality scores dropped dramatically. Okay. So basically, this is sort of what you'd look at. And I would say that this then becomes a questionable data set, because you'll see that, if you look at 20, you'll see that basically from about, let's say from about here, so that's about 100 base pair position 112 to the end of the read, we have a lot of our reads just showing low quality. So basically for half of the read length, we have low quality calls. So, this is something that's questionable. So this is where it becomes a bit tricky on assessing your data. So obviously you don't want to just throw data out, but what we can do is try and clean up data in a way whereby we can, for example, if we still wanted to save this data set, this particular sample, we might say, okay, well, I can cut the read, say at around 110. I'm going to have shorter reads. That is going to affect, depending on what you're trying to do with the reads, it's going to affect, your downstream analysis. So you need to weigh up what the effect is on your downstream analysis by cutting the read, or whether it's worthwhile just to remove the sample completely. So those are the things that you do need to consider if you are, want to clean up your data. I would always say that it's better to chuck out a bad sample rather than keep it in and get questionable results. Because at the back of your mind, you'll always know that you had that sample that was questionable. And then you might get results that are, again, become questionable because you've included that sample. So I would always say, rather, if you can afford to do it, just remove it. Even if you can't afford to do it, rather just remove it in order to get good quality data in the end. Okay. So that's the per base sequence quality. Okay. The next metric that we have is per sequence quality scores. Okay. So this is basically saying for overall, for all your sequences, what is the average quality score that's been observed? Okay. So on the left-hand side here, you have the number of reads, okay? And the plot is basically at the bottom here is the mean sequence quality. So it's looked across, so example, you've got 40 base pair reads or 40 base reads. It will calculate a mean quality score across those 40 bases. And this is what, so for each read, it's calculating a mean score, and then it's plotting the number of reads that at a particular mean quality score. So basically here we're saying that, what is this, 106, this is 160,000. In this particular data set, you'll see that majority of our reads have a mean sequence quality score of around 34, 35, 36, 37. So you'll see that majority of our reads have a mean sequence quality of phred score of around 37, 38, okay? And that's essentially, it's a good thing. Our mean quality score above 30, remember we said, that's a good thing. What we'll see here is this is an example of questionable data. You'll see the sort of what we call a bimodal graph. So you'll see that we have a reasonable amount of our reads, in this case, 60,000 of our reads, in the region of 27 to 32, 33. Okay, but we also have this bump here where we have about 10,000 of our reads with a mean sequence quality phred score of around 16 or 17. Okay, so this again is something that would be a red flag to note. And what we can do then, so for example, for this particular data set, what we can do is when we trim our data, we can actually say, I don't want to retain any reads that have a mean sequence quality score of less than 30 or less than, say, for example, 25, 26. And what will happen is through the trimming, we'll remove these ones that obviously have a mean sequence quality score of around between 14 and 19. It will remove these reads, okay? So this is one way of interpreting this output. So this is the per sequence quality score. It's an overall mean quality score value for all your reads in your data set. Okay. See, there's a question. So the sudden peak at the end of the graph is what we expect. Sorry, I'm going forward. So remember, these are two different data sets, right? This is an example of a good data set. So in a good data set, we expect to see this one peak where a majority of our reads, so here 160,000 of our reads are falling, have a mean sequence quality score of between, say, for example, what is it, 36 and 38. So usually we only expect to see one peak, okay? So in this instance here, we would expect to see this one peak here at the end. which is tending towards the more higher scores, so between 20 and 30, we would expect to see just this one peak. So this is saying that majority of our sequences in our FastQ file have a mean sequence quality score of between 25 and 33, okay? That would be this peak here. We don't expect to see this one, but this one is saying that we have a significant amount of reads in our data set where the mean sequence quality score is not anywhere near 30, it's actually between 14 and below, it's basically below 20. So this is basically showing that we have a lot of low quality reads in our data set. So this is what we expect to see and what we should see in our data set. And this is the data set that's where something strange is going on. So that's why you're seeing these two peaks. Hopefully that answers that question. Okay. So this is the per sequence quality score. The next bit we have, the next part of the report is the per base sequence content. Okay. So what this is doing, it's looking, so it's on the y-axis is essentially a percentage from 0 to 100, okay? And on the x-axis again is each position in our data set. So each position. So again, what it's doing is looking at each position in our data set, looking across all our reads, right? Looking across all our reads. And now it's looking at the actual content. So what bases are we seeing at a particular position in our reads, okay? And what we're expecting here is that across all our reads, we should see basically a consistent, relatively consistent percentage of A's, C, T's, and G's, right? Across all our reads, right, for a particular position. Okay, and that's what we're seeing here. So we're seeing a relatively consistent percentage of T's, relatively consistent percentage of A's, C's and G's as well. So we're seeing this sort of almost straight line across, going across our bases, right? And the reason for this is that, we expect there to be no bias in terms of particular bases at a particular position, okay? And if we do see, if that is happening in our data, what happens is you end up with this more sort of jagged looking lines. So where we seem to have over-representation of a particular base at a particular position, okay? So sometimes if your data is noisy, this will be observed. So you just need to be a bit careful about how you interpret this graph. You might also find that there might be a bit of noise at the beginning and the end of the sequences again. So just keep that in mind. So for example, here you'll see it's very noisy at the beginning. And that's just because these bases are usually not accurately called. As I mentioned before, we usually have low quality calls at the beginning, and it sort of evens out as you go through the read. So essentially what we're looking for here is, consistent smooth lines at this reasonable percentage of between, well, depending on your organisms, this might change, but this is sort of a reasonable region. I think this is human data. So it's a proportion of each base position for which each of the 4 normal DNA bases have been called. We're looking for this sort of straight line. If you do see anything like this, then there might be something strange going on in your data that you might need to look at in a bit of detail and look at it further. Again, I wouldn't be too worried if you see a bit of noise in these graphs. If there's something really dramatic happening, then there might have been some error in your library preparation where you have, you had certain parts of the genome were overrepresented. So you might have been sequencing parts of the genome multiple times. So this is just an indication that there might have been something going wrong during the library preparation step. Okay. The next metric that we'll have a look at is the per sequence GC content. So this is the percentage of Gs and Cs across your entire length of your reads. And what the FastQC does is it compares it to a normal distribution. The normal theoretical distribution is shown in blue in the graph. Okay, so here's blue here. And your actual GC content will be plotted in red. So again, on the x-axis, you have the mean GC content. And this is across all your reads. And essentially what we're looking for here is a relatively smooth normal distribution. So we're looking for a distribution that is similar. Let me just finish this and I'll go back to the question. We're looking for a distribution that is similar to the normal distribution. This again can change or can alter slightly depending on what sequence you are, what organism you're sequencing. We know that some organisms have different GC contents. So you just need to be aware of that. If you are sequencing an organism where you know the GC content is very, very different, you might want to then consider ignoring this metric because it'll show you, it won't fit to the normal distribution. It'll show you some strange results. So if you do have, regions, if you do know that your GC content is different, just keep that in mind when you are analyzing this particular output. So here's an example of what we would look at as being a reasonably questionable distribution for GC content. You'll see that we have, it follows the normal distribution, but it goes really high up at particular positions and comes back down. So this is, again, could be an indication of library contamination, sequencing contamination, or species contamination with other organisms. But this usually has something to do with the actual library preparation and not the actual sequencing experiment. So again, this you can view, but you do need to interpret it together with what you know about the organism that you're sequencing. There was a question about, can the quality of reagents contribute to the background noise? So in any of these that I'm mentioning where it could be due to library preparation errors, there's definitely a chance that your reagents could be an issue at that point. In most instances, it's not the reagents unless you know that your reagents are bad. In most instances, it does have something to do with, for example, the actual DNA quality or something major has gone wrong in the library preparation where you do have contamination. So instead of having just DNA from one sample, you've got DNA from a sample, your sample plus maybe another sample, or you've got contamination with another organism's DNA. But it can also be due to reagent quality, especially if you're getting consistent results that are bad, and you can't sort of narrow it down and you're using different DNA samples, then it could be possibly due to the reagents that you're using during your library prep. Okay. So the next metric that we're going to look at is per base N content. So this is again looking across all your reads at each position, and it's identifying if there are any ends in our reads. So ends in the read are basically where it's the instrument has not called a base. Okay, so it was unable to call a base at that position, so it inserts an N in our read. Okay, so essentially what we want to see is minimal amounts of N in our data, right? We don't want, at any position, we don't want to have lots of N's. So essentially in good data, again, this is percentage on the left-hand side, on the y-axis, on the x-axis, we have, again, each of the positions, so from 1 to 40. And what is plotted in red here is the percentage of N's. So at each position, it's looking for N's. And basically, we want to see this as close to 0 as possible across all our reads. So we don't want to see any ends. We don't want to see, basically, we don't want to see any ends at any of those positions. Here's an example of something strange that's going on. So between positions 25 and 31, we see that there's about 5%, about 5% of our reads are showing, we're seeing N content in about 5% of our reads. And this just could be something to do with when the sequencing was happening with the samples, something was going on at this position. It could be library prep that, for example, something was going strange happened with this particular, these particular fragments, where we had lots of N's being called. Well, not lots of N's, but essentially for this region, you'll see that there are N's in the data set. So again, yeah, you can be very strict, or you can be, more lenient, depending on whether you want to save your data set or not. But essentially, if you look at any data set and it's got lots of N's in it, I would just chuck that data set out. This one, I would say it's acceptable. You could still, I would still run with this data set. But if you like it says, yeah, if you see N content greater than 20%, then I would say, okay, well, maybe it's not worthwhile. And that something strange probably happened with this data set. So we probably might as well just chuck it out. So again, you can be, you can decide what your cutoffs are and how liberal or strict you want to be with N content. There was a question here, does the maximum DNA recovery increase the quality of per sequence GC content? I wouldn't say that would be true. If I understand it correctly, that's the maximum DNA recovery. Let me try and get back to that. I just need to think about that a little bit. Let me see. I'll try and get back to that at the end of the lecture. Just remind me if I forget. Okay, so the next metric we're going to look at is sequence length distribution. Okay, so basically this graph is telling us about what is the sequence length of most of our reads. So if you remember and it's giving the distribution of the lengths. So remember at the beginning of in our summary, we saw that our read length was 40 base pairs, 40 bases. Okay, so essentially what we expect in our particular data set is that majority of our reads should be at least 40 base pairs, okay, 40 bases. We will have obviously some that are less than that and some that are more than that. Okay, that's just the nature of the experiment. We end up with some reads that are longer, some reads that are shorter. Okay, so what we should see for this particular plot, we should see that for majority of our data sets, our read length is 40. And this is exactly what we see here. So remember, we had 250,000 reads. Okay, and you can see that majority of our reads, so this is our distribution, majority of our reads fall between 38 and 41 base pairs. Okay, so this is our distribution. Majority of our reads are within this 40 bases. Okay, so that's what we expect to see. Okay, and again, here we're expecting, this is expected based on what we expect to see, what we know our read length should be. So if we knew we were generating 150 base pair reads, essentially our distribution should be, between 140 and 160, for example, or 145 and 155. So we should see that sort of similar distribution based on what we expect our read lengths to be. Okay. So this is good. This is what we expect to see. If we see, this is another example whereby we have most of our read lengths are between, what is this? It's a bit difficult to read. So here we have between 0 and 1,200. If we look here, this is about 6, 7, 800, about 800. So again, this is just reiterating the point that this is based, what you see in this graph is based on what you're expecting to see in terms of your read length. And this is an example where we have varying read lengths that range all the way from, say, for example, 100 base pairs all the way to 700 base pairs, okay? So this is an experiment where we don't only expect to see read lengths of mostly around 40. This particular sequencing experiment has a read length range of between, for example, 100 and 800 base pairs. Okay, so in order to interpret this graph, you need to know, how your library was prepped and what is the expected read length that you will observe you're expecting to see in your FastQ file. Okay. The next metric, I think we're almost at the end. Let's look at the time. The next metric that we'll have a look at is sequence duplication level. Okay. And what we're expecting to see here is that we expect, so when we are doing for example, a whole genome sequencing experiment, remember that we fragment our DNA and then we generate a library. We might PCR up our fragments. We generate a library, but we essentially expect that each of those fragments will be unique. And that although we, in a sequencing experiment, we might have these, when we're doing the sequencing itself and reading off the sequences, we PCR amplify the fragments, but we read off one read from each of those spots. So what we expect in our data set is that there should not be fragments that are exact duplicates of each other, okay? Remember, we fragment the whole DNA, we then PCR amplify, and we PCR amplify so that we can read off the sequences. But each of those fragments should be unique, right? There should be some overlap, but they shouldn't be identical duplicates of any of our reads. And if we do see that, then we know that there was probably something strange going on in our PCR amplification step or our library preparation step, or we duplicated fragments when we were actually sequencing after the PCR step. So what we expect to see is very low or no levels of sequence duplicates in our reads, okay? And this I'm speaking for example of whole genome sequencing or exome sequencing. There is a slight difference when you're looking at transcriptome data, RNA-seq data. You might find, you probably will find some level of duplication in RNA-seq data. So again, you need to make sure that you understand what your data set is, what your library prep was, what experiments you ran, what sequencing you did, what your biological question is when you are assessing these FastQC results to make sure that you do it accurately. Okay, so basically what we see here is percentage of duplication, okay? And what we want to see is essentially a graph that looks like this with very low, this is sequence duplication level, so 1 to 10 in this case. And what we see here is that basically we have very low levels of any duplicate sequences, okay? So we have one copy or one fragment in most of our data sets that's unique. We don't have any where there's 2, 3, 4, 5, 6, 10 copies of a particular fragment, okay? So for this is an example of sort of bad looking data where you see that we do have some percentage of 2 copies duplicated, So this is about over 20% of our data set has, there's over 20% of our data. There's 2 copies of a particular fragment. It goes down, and then you'll see again here, towards the end, almost 30% of our data set has fragments where there's 10 copies of duplicates. So this is exact duplicate sequences. Okay, so again, this is indicating that there could have been something going on. There was certain fragments that we amplified multiple times, exact fragments that we amplified multiple times. So again, this can be used to look at the quality of your sequence data, and also the quality of your PCR amplification, your library preparation as well. Okay, so essentially the bottom line is we don't want to see a significant amount of duplication. Sequences that are duplicated in our data set. If you're looking at whole genome, exome sequencing, targeted sequencing might be a bit different. Transcriptome sequencing is very different. Okay, you might have some level of duplicated sequences. Okay. Then the second last one is overrepresented sequences. So again, this is kind of similar to the duplicated sequences. But this is where you have a particular sequence or read that occurs multiple times in your data set. So if that does happen, again, we don't expect to see this. But again, depending on your biological question and your experiment setup, this might be what you're looking for. So for example, one example that I do know of that makes sense is there was a study where we were looking at identifying transcription factor binding sites. So we had this experiment where we had our fragments of DNA, we mixed it with the transcription factor that we expect to bind to the binding site. We then pulled down all the DNA where the protein was bound. We separated the protein from the DNA, and we then sequenced those fragments, right? When you run that through QC, we expect to see an overrepresentation obviously of the sequence that the protein bound to. So when you looked at the FastQC results, you actually saw that there was a high percentage of sequences that were overrepresented. But that in that particular experiment, that's exactly what we wanted to see. Okay, so it's an example where how you interpret it is based precisely on what the question is that you're asking. If we're doing, for example, whole genome sequencing on a human genome, and we know that there's nothing different about this genome, it's just a normal genome, we don't expect to see overrepresented sequences, or we don't expect to see high percentages of a particular overrepresented sequence. Sorry. And the reason for this is that, again, we don't expect to see duplicate sequences, so we shouldn't see overrepresented sequences. And when we do see overrepresented sequences, I've seen it in data sets. It's most likely to be adapter sequences from your sequencing experiment. It's most likely to be primers that have been left in your data sets. Yeah, so I've mostly seen adapters and primers. So that's some reasons why you'd see these overrepresented sequences. And if you do, and it's a relatively high percentage of the total of your number of reads, then you do need to look into it and see, what was the source of these overrepresented sequences. Was it some sort of contaminant? Is it an adapter? Is it a primer? Okay, so you do need to investigate it if you do find a high percentage of these overrepresented sequences. Okay, and then the last metric is something called KMA content. Okay, So essentially, this is a metric that looks across your positions in your base, in your read. So again, you have percentage on the y-axis, and then we have position on the x-axis. Okay, so at each position, it's looking and it looks for overrepresentation of particular k-mers. So k-mers could be, for example, these particular basically this is based on what it observes in your data. So it looks for overrepresentation of particular sequences, short sequences in your data set. And what you expect is, so for example, if, for example, position 1, 2, 3, 4, 5 had some primers still stuck to it in terms of your reads, you'll see something like this happening at the beginning, where you have a very high representation of particular k-mers at a particular region. And that's usually the case where you see if you have, for example, primers still attached to your data. And essentially what you want to see is you want to basically not have any overrepresentation of a particular k-mer in a particular region of your read, okay? Again, unless you're expecting to see this based on the experiment that you are running. So again, you just need to interpret this again based on what you're expecting to see. This can also get a bit messy. I tend not to use, or maybe I shouldn't say that, but I tend not to use these results too strictly, unless it's something very obvious. Like for example, this here, we're getting these similar patterns for all these k-mers across a particular part of our read. Then that's quite obvious. But it can be a bit misleading. So I would suggest that you identify or play around with this. Don't interpret this too much. In most instances, it will come up. I didn't mention this before, but when FastQC runs, if you get a tick, that basically means that your data looks good. If you get an exclamation mark in yellow, that means that it's in between. And if you get a red X, that means that based on what FastQ thinks, your data has failed that particular metric. The ones when it's in yellow, those ones you always need to just look at with a bit of caution. So this one I would say just look at it with a bit of caution as well. Oh, there's one more, and the final one is adapter content. So this is actually specifically looking for adapters in your read data. So FastQC has a plain text file which contains the sequence for any adapters that are used by the different sequencing companies. And this is obviously updated on a regular basis. And what it does is it looks through all your read data and it tries to identify if there are any of these adapters in your sequences. So what you'd find is if there is adapter content in your data, it will usually also come up as an overrepresented sequence in your overrepresented sequences output. And then it'll also come up in the adapter content. The only difference with the adapter content is that it's looking for specific sequences, so it knows what the sequences are, and it looks for those sequences in your read data. So in some instances where service providers are not doing their job very well, they will leave in some adapters, some primer sequences in your data sets. And then you will need to actually remove that manually before you start doing any analysis. So that's important. Again, here we have an example where there's nothing. So again, this is our positions in our read and percentage, and it's found none of these. For example, was looking for Illumina universal primer, it was looking for an 3' adapter, 5' adapter, transposon sequence from Nextera, solid small RNA adapter. It hasn't found any of those in our particular data set. Okay, so there are some tips for interpreting FastQC. So whenever you have a warning, so remember I said where it's yellow or failure, where it's an X in red, you should have a look at the data sets and determine whether it's worthwhile keeping the sample in. Also determine if there's an option for trimming the data to improve the quality. Okay. Again, there's a trade-off here between losing samples. I know every sample is very precious in most instances. So there's a trade-off between losing samples, but then also affecting the quality of the results that you get out in the end. Okay. So here's some advice. So for sequence duplication levels, you will sometimes get a low number of duplicated sequences. It is organism dependent, as I mentioned before. Higher number of duplicated sequences expected in transcriptomics, so with RNA-seq data, so just to be aware of that. Overrepresented sequences. So for small RNA libraries, you will find that there might be some overrepresented sequences. And there may be some sequences that are naturally present in parts of the library. So you do need to know about your organism and what to expect in terms of overrepresented sequences. And then KMA content I mentioned before, they are platform dependent. So you might find some fluctuations depending on what sequencing you've done. So again, go read up on the documentation and know, what to expect to see if there are strange, differences. And there are some typical artifacts that will lead to this KMA content occurring. For example, I said at the beginning of the reads, at the end of the reads. So these are things to be aware of when you are analyzing the FastQC outputs. I'm going to stop here. Any questions? Okay, so I'm going to go on. I don't think there's too many slides left. Now we're going to go on to, okay, we've run our data through FastQC. We've had a look at the results. Now I'm deciding now whether I need to, for a particular sample, do I need to trim it to improve the quality, and how do I do that? So if your data does show bad quality, you might still be able to save it. It's not like, oh, well, depending on what, where the problem is, you might be able to save it by filtering and trimming your data. And one of the major metrics that are used for, the quality of your data is the quality score. So going back to the first graph that we had a look at. So for example, in this one, you'll see that we have quite strange data going on here. So this is looking at quality scores. So this is the first graph. So this is the box and whiskers plot. And what you'll see is that for the whole data set, we have quality scores at each position ranging from 2 all the way to 34. But what you'll see is that majority, for the first part of the reads, majority are all sort of above 30 up until about position 25. Then it starts to drop to 20 around here. And we've got these long tails here. So we have samples in between that are dropping all the way to 2. Okay. So what we can do is we can use, some tool to say, I want to, for example, only keep reads with an average score of above 20 across the entire read. And we might be able to clean up the data whereby, we can go from something like this to something like this, which looks pretty good. We will end up losing some of our data, but then we at least are saving the sample, right? So we can filter data based, or we can filter and trim data based on various metrics. So we can use mean quality. So for example, we have 40 base pair reads. We can say calculate the mean quality across all 40 bases. and only keep the read if the mean quality score is, for example, above 30. Okay, so we can calculate all that, and we can have tools that allow us to say, okay, well, I only want to have reads with the mean quality score is above 30. We can use read length. So say, for example, we expect our reads to be 40 base pairs in length. Then we can say, I don't want to use any reads where the length is less than 30 or the length is less than 20. Okay, because we know that those are probably bad reads because we expect our read length to be 40, so we expect it to go from, for example, 35 to 45 kind of thing. That's the range. So anything less than 20, you know, is going to be bad quality. You can also say read. We can select reads based on read length after trimming. And I'll come back to this a bit later. We can look at percentage of bases above a certain threshold. We can trim out adapters. So we can say if we have identified adapters in our data set, we can use a tool to say remove all the adapters from our reads. And we can also remove all adapter reads that are just adapters as well. Okay, so this is how we can quality control our data. So there's various ways to correct bad quality data. So there's read trimming. So I mentioned this before, we can trim off adapters or low quality regions of the read. So for example, remember I told you that with most sequencing technologies, at the beginning of the read and at the end of the read, we usually have low quality bases. So you can use a tool to say, okay, well, I've looked at my FastQC data, and for majority of my reads from position 1 to 5, I have very low quality reads, very low quality bases being called. So for all my reads, chop off position 1 to 5. And also at the end of my read from position 35 to 40, it's also low. So for all my reads, chop off 1 to 5 and 35 to 40. And I only want to keep what's remaining from that read. Okay, so that's the tools that allow us to do that. One of the tools that we hopefully will be able to use in the practical is a tool called Trim-O-Matic. Then we can also trim based on fixed length, the quality score, which I mentioned before. So fixed length, so based on what you expect your read length size to be, I spoke about before. And then quality based, so only retain reads that have an average quality score of a certain value, which is like Q30. We can also combine them, so we can say, look across my reads. I only want to retain reads that have a quality score, a mean quality score of Q30, and they must be greater than a read length of 20 or 35. Okay, and then adaptive trimming, which is platform specific, which I spoke about before. Okay, so in summary, I know it's been a bit of a long lecture, but hopefully it's been useful. In summary, FastQ raw read files contain information about the quality of each base call. We need to quality control our FastQ files in order to make sure that our read data is of high quality, because if we start off with poor quality data, our results at the end of the day become questionable. FastQC is the tool that we can use to generate various quality control metrics for each of the FastQ files. I've shown you how you can interpret each of the FastQC metrics and assess them. Again, always keeping the biological context in mind when you are assessing them, to determine the quality of your data, and then also determine whether your data needs to be trimmed in order to improve the quality. And if the quality of the read data is low, there are tools that we can use to trim the data to retain only high quality reads. So that is all I wanted to speak to you about today in terms of the lecture. I'm going to stop sharing my screen. And I know that I have put together a practical, which you should have access to on the course website. I'm not sure of the infrastructure and the access to the files, so maybe Kennedy, you can assist with that. But I'm also, so maybe before I do that, let me just take any questions that we have, and then we can move on to the practical. You're welcome to raise your hand and unmute and then ask the question. Mishak, you can go ahead.
So, thank you. It's quite informative. What I needed to know is now, assuming you have your raw data, the FastQ file. Yeah. So there will be tools for now generating the FastQC.
So yes, so the FastQ files are actually input files that you will use. So the tool is called FastQC. So FastQC is the tool that you will use. The input file is the FastQ file. And then once you run the FastQ file through FastQC, you'll generate that report file that I was going through. So all those graphs, yes, that will actually be generated for each FastQ file. So FastQC is a tool. You run the FastQ file through FastQC and you'll generate that report.
Okay, so then that would also, just looking at in your initial introduction, looking at the whole pipeline. So from there, then we'll also get to the tool for doing the assembly and aligning and assembly and so forth.
Yeah. So once you've quality controlled your data, you'll end up with your FastQ files that have been quality controlled or trimmed if you decided to trim them. Yes. And then that will become the input files that you will then use for whether you do assembly or you do alignment or you're mapping back to a reference. So those then will become the files that you will input into the next set of tools for doing the alignment and assembly.
And okay, then the other question would be like, so assuming you're using probably the Linux yes, platform, so would you also need to then use these tools within Linux, or then you could still do it differently in Linux?
So majority of the...
So that is now, let's say, command line as opposed to then just subjecting, the data into the different softwares. So you'll still need the softwares within the Linux.
Yes. So you also have to, the software needs to be installed within the Linux environment. I'm hoping that that's been done for you and you have access to a cluster where this has been done. So the software would have to be installed and you'll actually run FastQC on the terminal, the Linux terminal, and you'll get the output files. And that you would have to view in a web browser. Yeah. But the whole analysis, I mean, I think everything that you're learning will have to be run on a Unix command line.
Okay, thank you. Thank you.
Great, thanks, Mashak. Ntengai, I think I'm pronouncing that right. You can go ahead.
Thank you, Alon, for excellent lecture. The question, I posted my question in the chat, but I think I didn't put it well. I was just concentrating on the quality of the DNA extraction. How does it going to affect the GC content? But now my question is in general, how the quality of the DNA extraction is going to affect the base calling in general. I mean, extraction, PCR, and then sequencing, and then the output of the data.
So that's a good question, and it depends on the protocols. So for most sequencing experiments, there is sort of a minimal concentration and quality of DNA that's required in order for you to yield a successful amount of sequence data in the end. So the quality and quantity of the DNA that you start off with definitely has a major effect on the quality of the sequencing that you get out at the end. if you start off with very bad quality fragmented DNA, you will get very bad quality sequencing. So it definitely has a direct effect on the quality of the sequencing that you get out. So for example, for short read sequencing, so for example, Illumina sequencing, the requirements are not very strict. It has to be very high quality DNA, and there's certain concentrations that you need to give as starting material in order to start sequencing. I know for long read sequencing, the newer technologies, PacBio, ONT, the requirement is very strict in terms of high molecular weight DNA, high quality, high concentration requirements in order to do long read sequencing. So definitely the quality and quantity of your DNA starting material has a very big effect on the quality of the sequencing that you get out. And usually, if you run FastQC, and you will find that for majority of those metrics that I spoke about, you're getting very strange results. It usually means that your starting material was very poor, or you didn't have a reasonable amount, high enough quantity of DNA to start off with, or your DNA was very fragmented. So that's a good question, and definitely you rather start off with good quality DNA, good concentration, in order to get good results, rather than wasting money on sequencing DNA that you know is very poor. So there are some tools, there are some protocols. So I don't work in the lab, but I do know that there are some protocols, excuse me, that you can use to improve the quality and clean up DNA. Some of the labs don't suggest this because it does affect the quality. So if you do have, if you do have, DNA samples that might be degraded, you can use some of these protocols to try and clean it up and improve the quality. But yeah, I would suggest always making sure, and most service providers will tell you, they usually check the quality of the DNA before they do the experiment. And if you have bad quality DNA, they'll just tell you, sorry, we can't do the sequencing because you're going to get bad results. So I hope that answers the question.
Thank you so much.
Thanks. So Emmanuel was asking a question. Are there any other quality control tools or software that generate reports significantly different from what was presented today? I'm not sure what you're getting at, so maybe you can just, if you want to just explain it further, that's fine. I did mention that there is a set of tools, SAM tools allows you to generate some quality control metrics on FastQ files. It does give you some, obviously in a very different way. So you can try and have a look at SAM tools, looking at quality control. It's also a lot more involved because you have to run SAM tools on the command line and then generate individual graphs. I guess FastQC is used as commonly because it's very simple and it's quite straightforward to use. So hopefully that answers your question. If it's something more particularly you want to know, just ask me, or let me know. Dennis is asking, can multi-QC be used for quality checks? So multi-QC is a visualization tool. So you'd have to run your FastQ files through FastQC first, generate your reports, and then you can use multi-QC to basically combine the results of all your FastQC runs and view them in some very good graphs so you can look across multiple samples at the same time. So multi-QC is a visualization tool for results that have been generated by FastQC. So you'd still have to run FastQC first, and then you can use multi-QC to visualize those. So I showed you FastQC output. Those graphs that I showed you are from FastQC. So what multi-QC does is it takes those reports and it tries to plot all those results for multiple samples in one single visualization. So you can look across multiple samples at the same time. Okay, so it's quite useful, but it's just a visualization tool for results that come out of FastQC. So people are asking about the recording. I'm assuming the recording will be available. Yeah, Margaret says yes. Any other questions? Okay, if there aren't any other questions, I'm going to ask Kennedy or Margaret, what is the plan for the practical session?
Yes, thank you very much, Shaun, for the insightful presentation on NGS QC, the quality control and assessment. So yeah, there is a practical session after this presentation going to take place. We will have a break about 10 minutes, then we'll have the practical session up to right now it's 10:32 a.m. East African time. I'll have the practical session running up to 12 midday. Yeah.
Okay.
So yeah, the accounts are available. They have accounts in the ERI server.
Okay.
So tools are installed. They'll just module load the tools.
Okay.
The data is also available. I saw it was uploaded in Canvas, but for easier, it is available on Google Drive, but for easier access, we have it shared using a link. It is also on the server, so we just get the data, and each of the participants will have it on their end.
Okay, great.
So we can have a short break up to 10:40 a.m. East African time. Then we'll come back and begin the practical session.
Great.
Okay. So if you have, yes, go on.
Sorry, Oscar's got a question. I said I'll answer it when we get back.
Yeah, sure.
Great, thanks.
Okay, see you guys at 10:40 a.m.